Hello, Biology 158, and welcome to our final chapter for this summer semester on the reproductive system. So we'll go ahead and get started. So with the reproductive system, we have four major functions. So the first function is called gametogenesis. This is just the production of gamete cells, so genesis being beginning and gameto meaning gamete. So we're producing gamete cells. Gamete cells are sperm cells in males, and then oocytes in females, which are also known as eggs. You need both the sperm and the egg to make a zygote, which will develop into an embryo and then a fetus and then a baby and everything. That process is fertilization, the second major function of your reproductive system. When that sperm ultimately reaches the oocyte and manages to break through the outer barrier, those nuclei will fuse and then again make a zygote. Additionally, the reproductive system of the female is responsible for developing and nourishing the newborn. So with the placenta, all the nutrients will be provided to the fetus. Eventually, they will deliver the baby, and then afterwards, female hormones will produce a lot of milk through the mammary glands for postpartum nutrition. Then we have, lastly, the production of reproductive hormones. So the obvious ones we have are estrogen females, and then testosterone in males, which largely contribute to primary and secondary sex characteristics. But we also have lesser known hormones, like your follicle stimulating hormone and your luteinizing hormone, that are going to contribute to the initial production of testosterone and estrogen. So with the production of sex cells, or gametogenesis, the process by which they are formed is called meiosis. And many of you may have covered meiosis when you talked about introductory biology courses. You learned about mitosis, which is just cell division all throughout the body with any other type of non-sex cell. When we talk about sex cells, we are talking about the process of meiosis. And this is when we have a diploid cell, or a cell that has two sets of chromosomes of 23, meaning 46 total, and we form a haploid cell, which is just one set of chromosomes, which is just 23 chromosomes. So meiosis is broken down into two major cycles. So we have meiosis 1 and meiosis 2. You're not responsible for the individual steps of this. I just have the diagram on the right to show you the overall process. What I need you to be aware of is that meiosis 1, that very first cycle, is all about reduction division. So that is where we reduce our diploid amounts to our haploid amounts for cells. So when we start in meiosis 2, we have a diploid cell, but when we finish, we have two haploid cells. During meiosis 1, we have a very special event called crossing over. This is when we have different pieces of the chromosomes break off and exchange with other chromosomes to exchange genetic information. This is why all of the sperm cells and all of the egg cells are not genetically identical. They're all different because they have different sequences of DNA based on that crossing over from different pieces of the chromosomes breaking off and moving. Then we have meiosis 2, which is in this right column, and this is just similar to mitosis, where now we're just producing cells that have the similar number of chromosomes. So in this case, we have haploid cells, and they're just going to haploid cells. But in this case, we'll end up going from 2 cells to 4 cells. So meiosis 1, all about reduction division, going from diploid to haploid. Meiosis 2, all about going from 2 haploid cells to 4 haploid cells. Okay, so we're going to start with the male reproductive system and go through piece by piece. First thing is we are going to start talking about some of the more external structures. So we're going to start with the scrotum. The scrotum is just basically a big chamber that holds your testes, which ultimately produce your sperm. And there is a big separative structure called the raphe. The raphe is just this midline ridge that separates one chamber from another. Within the scrotum, you have 2 special muscles. They're called the dartos muscle and the cremaster muscle. And these are responsible for helping with temperature regulation. So what happens is that scrotum holding those testes are outside the body. And when it gets too cold, those dartos and those cremaster muscles are ultimately going to contract. When that happens, the scrotum is going to shrink in size, and that scrotum is going to be brought closer to the body, ultimately warming up the testes if they are too cold. On the other hand, if it gets too warm, that dartos muscle and those cremaster muscles are going to relax, and that's going to increase the surface area and space of the scrotum, so dissipating more heat, and the scrotum is going to move further away from the body, ultimately cooling it down. So there's going to be sort of a feedback loop in terms of contracting or relaxing based on the temperature needs of the testes. Then you have another structure called the perineum, and this is basically just the entire outer diamond shape between the thighs, but also between the penis and scrotum and the coccyx muscles, or sorry, coccyx bone of the spinal cord. Sorry, not spinal cord, of the spinal column. So with this diagram, we have to show you the initial structures. We have our scrotum here, which is the giant sac-like chamber that contains your testes where your sperm is produced. And then you don't see it, but we have within the scrotum lining, we have those dartos muscles and those cremaster muscles, which will contract to bring the scrotum closer to warm up the testes, or relax to get the testes further away from the body to cool them down. Then with the perineum, it's broken down into two structures. First is the urogenital triangle. This is the area that's considered between the pubic symphysis. So the pubic symphysis is that area of cartilage that holds the left and right pelvic bone together. So that includes the penis and the scrotum, and then it runs all the way to the ischial tuberosity of the pelvis, which is also known as the bony part of your butt. And all that area is the urogenital triangle. Then the second part of the perineum is the anal triangle, which runs from that ischial tuberosity on both pelvic bones all the way to the coccyx of the spinal column, and includes the anus. Next, we have the testes, and these are the actual organs where the sperm themselves develop. The testes are broken down into exocrine and endocrine functions. So your exocrine is all about secretion. This is where your sperm cells are secreted out of the testes. And then you have endocrine cells that produce testosterone, the major hormone for male sex development. On top of the testes, you have what's called the tunica vaginalis. This is a serous membrane that is both on the surface and just outside the testes themselves. Then you have a large piece of connective tissue called the tunica albuginea. And this structure ultimately divides the testes into lobules, or smaller sections. Then within the lobules, you have a bunch of scrunched up tubules called the seminiferous tubules. Those seminiferous tubules are where sperm is actually produced and made. Then wrapped around those tubules, you have what are called interstitial or Leydig cells. I'm always going to refer to them as interstitial cells, but Leydig cells is also appropriate. And those cells are all ultimately responsible for secreting your hormone testosterone. So within the tubes themselves, like I mentioned, the seminiferous tubules produce the sperm. And when the sperm has been produced, they are going to move to another structure just outside the tubules called the tubuli rectus. Think of these kind of like the minor calyces within the kidney that ultimately come together to produce the major calyces. And in this case, all the tubuli rectus will form a mesh network that's called the ready testes. And the ready testes will then move through efferent ductules to a structure called the epididymis, just outside the testes. So here we have a sagittal section of the testes themselves. We have that thin connective tissue here called the tunica albuginea, and that divides the testes into your lobules, all these little pockets that I'm circling right here. And within all these pockets, we have your seminiferous tubules all scrunched up here, which we have a histology slide over here, have all the production of your sperm cells. So all these cells that are lining the tubules are your sperm cells. Then, once these sperm cells are developed, they will move into the tubuli rectus, which is in front of each lobule that I'm moving my mouse over. And then all those tubules the tubuli rectus will all come together into the ready testes, that mesh network, which will then leave the testes through the efferent ductules into the epididymis. Then if we look now more specifically within the seminiferous tubules, we have a cross section right here. And this is where we have all of these sperm cells being developed. And then surrounding the actual tubules, you have your interstitial cells. So again, these are responsible for producing your testosterone. Then within the tubule itself, you have a second type of cell called the sustentacular cell. These are embedded within the walls themselves, and these are responsible for providing nourishment for the sperm cells as they grow up and develop. The sperm cells themselves are divided into three major sections. So you have a head, you have a midpiece section, and then you have a tail. Within the head, you have a special structure called the acrosome. This is the little cap on top of the head. That acrosome is responsible for producing digestive enzymes. And those digestive enzymes are going to help the sperm dissolve the outer covering of the egg so they can burrow in and try to fertilize the egg. Like all cells, you have a nucleus responsible for all of the functions and responsibilities of the cell. Then for your midpiece, you have a giant section here that has a lot of mitochondria in it. All that mitochondria, or rather most of that mitochondria, goes into powering the tail, which is a flagellum. That flagellum is also just generally called a whip-like tail, which helps propel the sperm cell down the reproductive track, ultimately towards fertilizing egg. So the testes themselves don't actually start outside the body. During a boy's development, from birth until puberty, those testes generally make their way from the abdominal cavity down to the scrotum. So ultimately, they start in the abdominal cavity, and they're going to start to move through what's called the inguinal canal. The inguinal canal is just the area between the anterior abdominal wall and the bladder. They're going to move through a specific pathway that contains what's called inguinal rings until ultimately they reach the testes, as shown in the diagram here. They're guided by a special structure that's called the gubernaculum. Ultimately, that gubernaculum will develop into a scrotal ligament, but they're ultimately what's guiding the testes from that abdominal cavity to the scrotum. If this process fails, you get a condition that's called cryptorchidism. It's always a mouthful to say. This is when you don't have a testes or both testes make it to the scrotum. It fails. So they're still stuck in the abdominal cavity or partway down. If this happens, ultimately you're not going to have normal sperm development because the testes are not where they need to be, both in terms of a regulatory environment, but also in the position they need to be in to receive signals to produce sperm. And the process by which sperm is produced is called spermatogenesis. So spermatogenesis is just a type of gametogenesis. As the name implies, this implies the production of sperm in, again, the seminiferous tubules. So a couple of different steps that we start out with, roughly 5. The most precursor cell you have to a sperm cell is called the spermatogonia. These are also what's known as your germ cells. Those are going to divide by mitosis. So we're just making more copies. We're not doing any crossing over or reduction division yet, into some daughter spermatogonia, so we can produce more of them. And then ultimately your primary spermatocytes. Your primary spermatocytes then divide by meiosis, the first meiotic division, to produce your secondary spermatocytes. After a second mitotic division, you then have your spermatids. Then you have the process by which those spermatids become sperm cells, and this is a process called spermiogenesis. This is where the sperm cells actually form a head, as well as the acrosome and the midpiece and the flagellum and everything they need to function as a sperm cell. So a quick thing I wanted to point out, spermiogenesis is the process of going from spermatid to sperm cell. Spermatogenesis is the entire process of producing a sperm cell. So sometimes students get confused, spermatogenesis versus spermiogenesis. Spermiogenesis is just specifically spermatid to sperm cell, spermatogenesis, entire production of sperm cell process. So once we have that sperm cell and it's developed with the head and midpiece and flagellum and everything, you then have your sustentacular cells provide nourishment for those sperm, as well as produce that blood-testis barrier for the production of hormones. Because your sustentacular cells also convert that testosterone, that primary sex hormone, to dihydrotestosterone and estrogen. And ultimately, that's going to lead to the formation of more sperm cells. So it provides a positive feedback for that. Okay, so moving away from the testes, now we are into the ducts, which is the pathway for your sperm and other fluids. First duct we're going to talk about is your epididymis. This is just right outside the testes. And this is ultimately where your sperm are going to develop into their most mature state. So with the head section, the body section, and the tail section of the sperm. The epididymis has a special structure called the stereocilia, and ultimately these are going to help with absorption of any excess fluid within the ducts or the lumen of the ducts, so any sort of, or the space within the ducts, any excess fluid that doesn't need to be there during the sperm development. Then as we move from the epididymis, we get into the ductus deferens, also called the vas deferens. And ultimately, these bring the sperm cells all the way to the abdominal cavity towards the prostate. And there's a couple structures within the ductus deferens. So first one is the spermatic cord. So if we look over here on the right, what I'm circling, we have a section here cut open. And your spermatic cord contains a couple things like your blood vessels, the testicular artery and the testicular vein, as well as all the nervous tissue with the testicular nerve. Then you have a structure called the ampulla of the ductus deferens. This is just where the ductus deferens widens out and allows for more sperm cells to flow through. And ultimately, your ductus deferens is able to move sperm from the epididymis up the abdominal cavity by peristalsis, that process you've learned from digestion. Then lastly, we have the ejaculatory duct. So the diagram here makes it look like it's at the neck of the bladder. Just for context, it is actually running behind the bladder into the prostate. So the ejaculatory duct is not the neck of the bladder. It is its own channel that is in the prostate behind the bladder. The diagram, you're just not able to see it from this front view. But ultimately, that's going to join the ductus deferens to your seminical vesicle. And it's then going to lead into the urethra, or lead into the prostatic urethra within the prostate. Next, we have your urethra, which is divided into three pieces based on what structure it's moving through. So first is your prostatic urethra, which passes right through the prostate, appropriately named. Then you have your membranous urethra, which extends to the perineum. So it's this very thin and small section of the urethra between the prostate and the tip of the penis. And then you have lastly, you have the spongy or penile urethra, which runs through the penis out of the body. Within the penis itself, you have special glands called urethral glands, which produce this sort of slick mucus compound to keep things lubricated. Then you have the penis itself. So the penis has two different functions. First is to assist with urination, since part of the urethra runs to the penis. And then the second major function is ultimately to inject sperm into the vagina through emission and ejaculation. Ultimately, your penile tissue has three types of tissue. You have the outermost tissue of the penis, which is called the corpora cavernosa, which is what I'm circling right here in purple. Then you have the corpus spongiosum, which runs pretty much the length of the penis, all the way from the root until the glans penis. And then you have the root of the penis, which contains two smaller structures, the bulb of the penis and then the crus of the penis. All of those structures are able to be engorged in blood, which contributes to a penile erection. And then with the glans penis, that's the section of the corpus spongiosum, you have an area that's called the prepuce. That is the foreskin that normally covers the penis at birth. A lot of times for many males, you'll have a surgical removal of that. That's called circumcision. Then at the very tip of the penis, you'll have the external urethral orifice where urine or ejaculate will be removed. So then next we have your accessory glands. So when we talked about the digestive system, we learned about the main pathway of food, and then we learned about accessory glands that provide secretions, so things like your pancreas and your liver. Accessory glands are the same idea here. These are secretions that are mixing with the sperm to ultimately form semen. So first you have your seminical vesicles, which provide the majority of the secretions for semen. So your seminical vesicles reside right behind the urinary bladder, and they empty into the ejaculatory duct, which runs right here. So this is a better view of the ejaculatory duct from the ductus deferens, in that it runs behind the bladder right here into the prostate. So again, the seminical vesicles empty into the ejaculatory duct, and they provide a specific secretion that contains fibrinogen that is very high in pH. If you remember when we talked about blood, fibrinogen is responsible for blood coagulation when you get a cut or a wound. In this case, the fibrinogen is coagulating the semen, keeping it clumped together. Then you have your prostate gland, which also produces a high pH secretion. And in this case, it contains clotting factors, which again help with clumping. And then another compound called fibrinolysin. So what fibrinolysin does is that actually breaks down the coagulation and breaks things apart. So what happens is when the semen ultimately is ejaculated, it will ultimately be very clumped due to the fibrinogen and the clotting factors. But once it's deposited into the vagina, that the fibrinolysin will ultimately break that up and allow the semen, or the sperm rather, to swim up the reproductive tract. Lastly, we have your bulbourethral gland. So that is this really tiny gland right here at the tip of the penis. This ultimately helps with mucus secretion and then produces a very high pH compound to help neutralize the vagina. So you'll notice that all of these compounds, so the seminical vesicle, the prostate gland, and the bulbourethral gland, all have very high pH compounds. And the point of this is to help counteract the very acidic environment of the vagina. So in order for more of the sperm cells to survive for a longer period, they have higher pH and all of these neutralizing compounds, so they can make their way further up the reproductive tract before succumbing to the very acidic environment. Okay, so all of these secretions from the accessory glands and all the sperm cells we have from those seminiferous tubules collectively refer all to that as semen. So sperm cells plus accessory secretions is semen. And the semen is deposited from the male reproductive system through two phases. You have first emission, which is when the semen moves into that prosthetic urethra. And then you have ejaculation, where it's forcefully expelled through the penis by peristalsis motions. And then, as I mentioned, after the ejaculate is removed from the penis, you have fibrinogen, and it's those clotting factors contributing as well, but fibrinogen gets converted to fibrin, which helps keep that semen clumped until you have fibrinolysin help break it up from the coagulation. And then once that occurs, that sperm is going to try to swim over the vagina to fertilize an egg.